plan on getting out of here on time because it's a shorter talk today. So welcome to the Friday Speaker Series. I'm Evan Deutsch. I'm the Program and Outreach Associate for the Center. This is actually our last Friday Speaker Series event of the semester. And so a big thanks to everyone who supported us throughout the year. Really appreciate it, and we hope you'll join us in the fall again. We've had some incredible speakers this year, and today's the perfect way to wrap it up with Middlebury's Jack Byrne, the Director of Sustainability Integration. Jack is the co-founder of the nonprofit Foundation for Our Future, where he oversaw a project to improve sustainability education in K-12 curriculum. He's also a founder and first executive director of the Riverwatch Network, an international NGO supporting community-based watershed conservation. Jack holds a BS in biology from Kent State University and a master's in environmental law from the Vermont Law School. And today's Jack is going to talk about biohacking and synthetic biology and the ethical and entrepreneurial implications surrounding the topic. We were talking about it at lunch, and I think we're really excited to hear more. So again, thank you all for coming, and please join me in welcoming Jack Byrne.
So thank you all for coming and hearing me formally present for the first time something that's been on my mind for quite a while, since my biology days at Kent State in 1979. 1970 was when the technology for sequencing DNA first became widely available and relatively easy to use. So we're quite that cat is long after that. What I wanted to do at the outset is just create a few caveats. One, I am not an expert in this topic. It's something that I've followed and paid a lot of attention to. So I'm bringing it more of a journalistic perspective to it. And then I'll add the other caveat that I'm not a journalist. So I want to try to give you a pretty good kind of overview of what is going on in the field of synthetic biology, kind of the spectrum of activity from big corporations doing huge commercialization, applying biotechnology all the way down to people we don't know what they're doing in their garages, and then in between. So, and I also want to relate it to sustainability and entrepreneurship, because obviously that's the topic that I devoted quite a bit of my professional and personal life to. And I thought it would be useful to look at John Ehrenfeld's definition of sustainability, which is the possibility that humans and other life will flourish on Earth forever. It's kind of an existential issue, and I think a lot of us share the same concerns that we may not be on a path that possibility remains a probability. And then the other notion of reducing unsustainability, which is a lot of what we do, will not create sustainability. That there are probably more fundamental changes in culture and psychology, kind of who we are, that need to happen. I'm not going to argue that synthetic biology will provide that fundamental change, but I'm going to suggest that it's a really important tool that can be used to help certainly reduce unsustainability and maybe even create it. And then Freeman Dyson, a physicist who makes predictions that are often right, said this about 10 years ago about the field of synthetic biology. So just want to try to cover these questions. What is it? How many of you have heard the term synthetic biology? How many of you know what it is? Okay, good. How big is it economically? What are the benefits and who's doing it? What are some of the risks and ethical issues associated with it? And I'll leave this up to you. How big a tool is it? So I'm going to turn 6 minutes of my presentation over to the government of Australia, who did a really great video answering this question, far better than I could do in a few slides. So let's hear their explanation of it.
After 10,000 years of genetic manipulation via selective breeding, humans finally gained direct access to the genetic code, deoxyribonucleic acid, DNA. Since then, we've cut and pasted it, photocopied fragments of it en masse, sped-read it with sequences, printed out the code letter by letter in the lab, modelled it on computers, and measured it with microscopes. For 40 years now, we've called this work genetic engineering. The trouble is that while there's been an extraordinary amount of genetic discovery and manipulation, there's been precious little engineering. Engineers are frustrated by genetics and molecular biology. The experiments are too slow, the complexity too messy, and growing more so all the time, and there's a frustrating lack of standardised components. They'd like to do to genetic engineering what engineers have done since the Stone Age: collect, refine, and repackage nature so that it's easier to make new and reliable things. Engineers want to treat DNA more like a programming language. Instead of ones and zeros, A's, T's, G's and C's. They want to use DNA to write simple, Lego-like functional components, inspired by, but not found in nature, and then run them in a cell instead of a computer. The only difference is this software builds its own hardware. They call this re-engineered genetic engineering synthetic biology. Nowadays, rather than cut and paste the DNA sequence out of one organism and into another, you can, if you know what you're doing, just type your DNA sequence into a computer, or copy it from a database, or even select from a growing component catalogue. And then you just order it over the internet. Yes, really. The DNA sequence may be copied from nature, but the DNA itself is made by a machine. It's synthetic. The raw material for synthesizing DNA is sugar, $25 of which will buy you enough to make a copy of every human genome on the planet. The chemical letters are fed into the DNA equivalent of an industrial inkjet printer. In goes your sequence information, and out comes DNA, at a cost of less than 40 cents per base pair and getting cheaper all the time. It's then freeze-dried and shipped to your door. Already engineers have assembled an open source catalogue of over 5,000 standardised components, called BioBricks. At an annual worldwide do-it-yourself competition, university students build new and more complex biobricks, string them together, and then run them inside a much-studied intestinal bacteria, E. coli. Sure, they're toy projects with shoestring budgets, but the results are impressive. E. chromi, an E. coli with sensitivity tuner and colour generators, is programmed to turn one of 5 colours when it detects a certain concentration of an environmental toxin. E. coloroid is a bacterial system which switches on and off in response to red light and acts like a bacterial Polaroid camera. Groups with more time and a lot more money are rewriting, or in computer programming, refactoring whole systems. Jay Kiesling, chemical and biological engineer, and his team at UC Berkeley have built and continually refined a new metabolic pathway in yeast by assembling 10 genes from 3 organisms in an attempt to produce synthetically the anti-malarial drug artemisinin, and to do it cheaply enough to treat up to 200 million malaria sufferers each year. Biotechnology pioneer Craig Venter has gone even further. His team has entirely replaced the DNA of one bacterium with a synthetic copy of DNA from another naturally occurring species, and added a few extras, like their email address. This wasn't creating life, it was testing just how reprogrammable a bacterial cell can be, an important step if we want biological factories which can be tasked to make many things, like vaccine, medicine, food, and even fuel. In the last 10,000 years, genetics has taken us from gathering seeds to manipulating DNA. And engineering has taken us.
I'm going to stop it there. It gets even more interesting in terms of where it might go, but we're far from that. So I hope that kind of gives you a better idea of what we mean when we talk about synthetic biology. So it's used in genetic engineering, as described in the video, in a couple of big areas, creating drugs for biologics. We're familiar with GMOs and agricultural systems, and then industrial processes to create different kinds of things like fuels and so on. And these are 2007 numbers. I just didn't have time to put in 2013. Rob Carlson. If you want a really good kind of start into this topic, this is a great book, Biology as Technology. He just updated these numbers for 2013. You can see they're pretty big. If you look at percent of US GDP in 2007, those three areas were close to 2% of GDP. He puts it at about 2.5% in 2013, and growing at the same rate, about 15 to 20% per year. So it's from a commercial standpoint, it's a pretty significant piece of the puzzle, or piece of the economic activity. So, who's doing it and what are they achieving? You kind of have this spectrum from, big companies like Monsanto, an interesting company I'll tell you a little bit more about called Amyris, Affymetrix, the whole range of kind of publicly traded, private, well-funded companies that are doing this. And some of them are producing some really impressive kinds of things at pretty large scales now. This company, Amyris, is kind of interesting. I don't know really well, and it may be not quite right to characterize it as the Ben and Jerry's of synthetic biology, but they have a really socially responsible mission. Their company really is kind of has very strong values around sustainability, around social responsibility, about doing good for people. One of the things that they're working on is essentially the piece you saw in taking the baker's yeast and producing artemisinin in a kind of, basically changing the metabolic pathway of the yeast to produce a certain output at efficiencies that are much better than a conventional refinery. They're doing those kinds of things. Producing biofuels from sugar. They've run 3 flights now over the last 2 or 3 years on biologically synthesized biofuel for jets. Just recently did one in January of 2014 for Etihad Airways, which is a UAE airline that will be bringing a lot of people to the World Cup, which they're trying to make a carbon neutral event. So there may be actually jets running people on biofuel into the Middle East. They've also worked with the Gates Foundation, received about 40 million, I think, to produce, basically to create a bioreactor yeast that will convert sugars into artemisinin, which right now is a very, well, not so much right now anymore, but it's a very difficult, slow process. They're using wormwood plants I think they're more prevalent in China, Chinese sweet wormwood plants. They basically developed a synthetic biology platform for doing this. Gave it to Sanofi, is that the Sanofi? What's the, Sanofi Aventis, to mass produce artemisinin to get to malaria. That's a malaria treatment drug. Sanofi is in the process right now of, I think they're producing large quantities of it. Not quite the amount they're looking for to really address all the cases of malaria that occur worldwide, but getting very close to it. And Amyris did it on a no profit, no loss principle. So I think that means you pay us for what it costs. We're not going to make a huge profit off of it. So, when you look at, how many of you are familiar with Moore's Law? You know, the cost of computing, you know, doubles every so often and goes in half at the same time. The same kind of thing is happening in the synthetic biology field. Lab instruments that you would see in a very elite university laboratory 5 years ago are now available commercially for pretty low cost. I'll show you what I found on eBay this morning. But you can see the price per base of synthetic DNA, the price per creating a pair, a DNA pair, has gone from tens of dollars down to tens of cents over this relatively short period of time. And then the productivity in synthesizing and sequencing DNA has gone up considerably in terms of the numbers of bases that are done per person, can be done per person per day, pretty significant increases. This is in comparison the number of transistors on a computer chip. Similar kind of productivity gain. So if you have a DNA sequence or a chunk of DNA sequence called an oligo that you'd like to have to do your experiments with, you can simply go online and order it. And it'll come in the mail, freeze-dried and ready to use. Or you can do it yourself. You can get a DNA synthesizer from eBay, you can get it from LabX, and you can get it pretty cheaply. This is this morning, eBay. DNA synthesizer, Applied Biosystems, $1,200. You need the reagents, you need to know what you're doing, but not hard at all to set up a garage Syn Bio Lab or DIY biotech, do-it-yourself biotech, or biohacking, which has a negative connotation. And we can talk about some of the risks. On the other end of the spectrum, who's doing it, we have now about 20 or so community biolabs that people have set up in their neighborhoods, in their basements, and have a kind of a community use of it. This one, GenSpace, in New York City, here's just from their website, an open barcoding night. Somebody went to Alaska and brought back samples of plants and animals, I suppose. And there's a barcoding of life project, which is a project to catalog the DNA of every species on the planet and have a database and a reader, so that if you're out in the field, you can take a sample of this plant or that animal, drop it in the reader, it'll read the DNA, look it up in the database and tell you what you're looking at. Or maybe that you're looking at something that, as far as the database knows, does not exist. So bring yourself and do some DNA reading of these samples and enter them into the barcode for life database, or at least enter them in that process. Ellen Jorgensen is the founder of the GenSpace in New York, and she did a TED Talk. And it's long, but I'm just going to play you a few minutes of it. She's talking about who's doing this. This may take a minute to catch up to the timestamp, but
so who gets to do it? I think we'd all be pretty comfortable with this guy doing it. What about that guy? In 2009, I first heard about DIY Bio. It's a movement that it advocates making biotechnology accessible to everyone, not just scientists and people in government labs. The idea is that if you open up the science and you allow diverse groups to participate, it could really stimulate innovation. Putting technology in the hands of the end user is usually a good idea because they've got the best idea of what their needs are. And here's this really sophisticated technology coming down the road, all these associated social, moral, ethical questions, and we scientists are just lousy at explaining to the public just exactly what it is we're doing in those labs. So, wouldn't it be nice if there was a place in your local neighborhood where you could go and learn about this stuff, do it hands-on? I thought so. So 3 years ago, I got together with some friends of mine who had similar aspirations, and we founded GenSpace. It's a nonprofit, a community biotech lab in Brooklyn, New York, and the idea was people could come, they could take classes, and putter around in the lab in a very open, friendly atmosphere. None of my previous experience prepared me for what came next. Can you guess? The press started calling us. And the more we talked about how great it was to increase science literacy, the more they wanted to talk about us creating the next Frankenstein. And as a result, for the next 6 months, when you Googled my name, instead of getting my scientific papers, you got this.
So, raising some of these issues around this technology. I think I said earlier, that cat's been long out of the bag. And it's not so much a question of if we deal with this, it's how do we deal with this. And that's an interesting approach to turn it into a community-based activity. And we'll talk a little bit about management and regulation of the technology, because there is a fair amount of it, but still pretty loose. Here is a list from DIYbio.org of the kinds of the labs that exist like GenSpace, about 20 of them in North America, most in the US, and a comparable number in other countries in the world, where the kind of stuff that GenSpace does takes place there. So this is kind of the entrepreneurship piece of this. If you look at the really significant advances in technology and in other economic areas that have occurred in the U.S., a lot of them happened in these small spaces, these garages, these community, efforts to try to solve a problem or create a better world. Here's Wozniak and Jobs making, return the personal computer into the personal computer. Ben and Jerry's, who made really good ice cream, made it popular. And a list of all the kinds of things that have been created by small companies, startups. And if you look at, this is really interesting, the net job creation in the U.S. over the last 30 years or so. Big companies, firms that are 1 year and older, they create a lot of jobs, but they also get rid of them. Yeah. They outsource, they offshore, they get more efficient. And if you look at the net of new job creation, a lot of it comes from these very young companies, the kinds of startups like the CSE, amongst, I'm sure, other parts of the mission are trying to help students and others achieve. So this is an area that also has a lot of potential from an economic and jobs creation standpoint. So there is obviously a lot of concern about, it's so easy to get your hands on this stuff. Equipment's pretty cheap. There's been a fair amount of study and commissioning done, but it still is, I think, relatively unregulated. And the bigger challenge comes, from a kind of startup standpoint, the commercialization aspect of this is a big barrier. Going from a good idea to getting commercialized, patent law around this is really complex. So that's one set of barriers, and then there's this whole, the potential for bio-error. You just kind of did something you didn't really intend to, or bio-terror, where somebody who's nefarious really puts their mind to doing something bad with it. So there's been some, you can't really read this very well, but this is an options for governance paper that was done by the President's Bioethics Commission, another paper, another report on making sure that we do everything we can to prevent these kinds of bad things from happening, or at least being better able to respond to them if they do. But the strategy for countering biological threats specifically acknowledges the beneficial nature of these synthetic biology efforts, including the kind of small-scale stuff that we've been talking about, what goes on in basements and garages and so on. So it's not treating it as something to be stopped and heavily regulated, but it is saying it's something we need to be concerned about. A whole set of recommendations here in terms of biosecurity, lab safety, protecting the environment. Again, I don't know the extent to which these are actually implemented, but my perception of this is that the regulation and management of this area is still pretty loose. And then, some of these more difficult questions. You start with the genome in nature, and you create something that's based on that, but it's different. Where do you draw the line about difference? And when does it become something we should be concerned about for security and ethical reasons? I think these are all questions that we're still grappling with. At the same time, we already have an effort going on to de-extinct extinct species, the Revive and Restore project. That is actually in the process, and I'm going to show you a little video clip of bringing back the passenger pigeon, which went extinct earlier part of the 20th century. Smithsonian, National Geographic, Longdale Foundation, a whole group of respected institutions have put a lot of money and effort into actually doing this. I'm going to play you a little video of that effort.
The passenger pigeon was around for 6 million years, and people remembered how amazing it was that these animals darkened the sky for hours at a time.
There's these stories about these giant flocks blocking out the sun, and their wings sounding louder than thunder.
We developed the techniques to hunt and net these birds and basically kill them all. And then it was really gone. When I was growing up, my mother said when the last one died in 1914, it broke America's heart. It was one of the prideful things that America had to show the world. It was gone.
This book, this is essentially the Bible of passenger pigeon. I am currently the lead researcher for the de-extinction project to bring the extinct passenger pigeon back to life. When I say, oh, we'll bring back a passenger pigeon, everyone goes, oh my God, that's like Jurassic Park. And no, it's not. Not at all.
Our scientists have done things which nobody's ever done before.
Yeah, but your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
I don't think a pigeon is going to, pluck lawyers off of toilets. So how we're actually going to go about de-extinction? Every organism has a unique code in their genes.
This would be good, I think.
So these are actual DNA codes from a chunk of a passenger pigeon foot. Now, if you can get DNA, then we can actually make it into a living cell. We're going to sequence its whole genome. We're going to build from scratch the code that is a passenger pigeon, one gene at a time, compare it to its closest relative. Then we're going to introduce DNA into a living cell of a band-tailed pigeon. And when you introduce an extinct animal's egg cell into a new mother, then you've changed the game.
So just one, I'm going to finish up here. So this has sort of come over into the conservation biology area just recently. About a year ago, this conference happened How will synthetic biology and conservation shape the future of nature? Synthetic biologists and conservation biologists came together to feel each other out and say, how do we deal with the power of this technology and the effort that significant worldwide effort to conserve and restore biodiversity. And I haven't seen the proceedings from this. I'm sure they would be really interesting. The questions they pursued, which some of you may not be able to read, just do it for you. I'm going to leave this talk, leave these questions with you to finish my talk, because I don't have answers, but I thought this would be a good launching point for a discussion. So, if synthetically produced species can replace extinct ones, then what is lost? When is a synthetically recreated species enough like the species it is designated to recreate, or it is designed to recreate? And how authentic? Are novel species that perform vital ecological functions supporting endangered species to be embraced or eschewed? This isn't quite on target, but one of the interesting things that somebody's working on is synthetically making ivory, elephant tusk, which of course means, because it's so prized in other countries, a lot of elephants get killed for their tusks. If you could flood the market with synthetic ivory, you couldn't tell the difference, maybe the elephants would be less pursued. And then is genetic variation introduced by a synthetic means any different than naturally produced variation, like mutations in nature? And would natural selection acting on these two different types yield different results? So I'm going to stop at that and ask your forgiveness for not providing all the answers to these questions, but give you a chance to share your ideas and questions. Yeah, Kevin.
Yeah, I guess one of the things for me that I was one concern that comes up about reintroducing extinct species and all of this in the passenger fishing in particular is that if you have an ecosystem that is not adapted to 100 years for passenger fishing, then you reintroduce them, and that brings another issue that may come about. What does de-extinction mean for how we've adapted since then? I mean, of course, I don't have an answer for that either, but it's another perhaps question.
Yeah. Yeah, I think it does raise probably the same kinds of questions that get raised anyway about what's left for these endangered species to actually live in. If we bring pigeon back whose habitat is it doesn't exist anymore, will it survive, or what will it do to other species? I know that there are a few places, and I think if I have it, if I'm remembering correctly, around Chernobyl there was a lot of land available that nobody wanted, and some of this land I think is now being used as a place to reintroduce some of the de-extinction is successful, to reintroduce some of these species that once occupied that region and give them that habitat to evolve in. And I guess in some ways it's sort of like a twist on a zoo. Yeah.
So species reintroduction, has that happened yet? That you know of it anywhere?
No.
Extinct species reintroduction?
Not that I know. I think this is the Reviving Restore is the first serious attempt, because I think the science is, the technology has reached the point where it's feasible.
Yeah, because my thought would be that even if they do manage to recreate basically a genetic clone of an extinct individual, then wouldn't all of they'd have to tamper with the DNA in such a way that they would be reproductively viable to create a new species. And if they just create clones, then that's not sustainable.
Yeah, I think that there are probably lots of really important questions that, like reproduction and proliferation. Still a long way to go, I think. Jeannie.
I would just kind of tag on to that question, just that some of the questions being brought up here are ones that conservation biologists are already dealing with and answering, just in the sense of reintroducing species that have been endangered to the point of only existing in captivity for many, many years. Or the question, like question 3, how are novel species, can they perform the same functions, basically? And a similar question that can be asked without even having synthetic species is a species from another part of the world that's not native okay for performing the same ecological functions? Like, for instance, the eastern hemlock is in danger of it's not endangered currently, but it's going to get wiped out across the east. And so the question is, if we bring hemlock species from Japan and put them across the east coast, will they fill the same ecological niche? So I think that there will be a lot of material already in the field of conservation biology to help answer some of these questions.
Yeah, in some ways, maybe the only difference is that we can do this. I think engineers can, rather than, I guess in a way we're engineering, if we bring the Japanese hemlock over and say, let's see how that goes.
Thank you, first of all. Second of all, I think it's really a question about my synthetic grandfather, right? At the end of the day, it's biology. And biology is big. I don't think we should be as necessarily concerned about trees or other species. I think there's a human component here that really is going to require diligence.
Yeah, we were talking about this at lunch. I think the Supreme Court has said you cannot patent anything that's created in nature. Okay, so do I own my genome? Which may have some incredibly novel but beneficial sequence that allows me to do something that nobody else can do. So I own my genome, and can I sell it? Can I be the copyright of the increasing the ability of blood to carry oxygen, become a super athlete, or somebody takes a copy of my genome and creates something else with it that's different, but based on that, who owns that? I think those are the legal issues around this are pretty significant as it evolves. And and then the question of should people be able to specify their offspring? What they want in a child.
Just a little bit more about the regulatory aspect of this, because it sounds rather questionable to be able to do whatever you want in your garage, and that's such great power with great power comes great responsibility.
Yeah, I don't know a lot about that, but I can say my sense of it is that there's not anything uniform and consistent. A few examples I know of people who've been accused of being biohazard creators and getting busted by the FBI. We have a speaker here, I didn't catch, who does art with synthetic biology. Did anybody catch that? Yeah, it was sometime this past year. And it wasn't the same artist, but there is a synthetic biology artist in San Francisco who was creating art using genetically engineered organisms, who got busted. Somebody said he's creating something bad, and the FBI at gunpoint busted into his house. But there is kind of an effort to regulate and track and prohibit the sale of the components of this kind of technology to licensed or registered entities, so that at least it's more difficult if you're an individual trying to do something bad to just get the stuff you need, and it can't be delivered to a residential address. It has to go to a laboratory with some legitimacy, whatever that legitimacy may be. And then one of the questions I have, which I don't know, is some pathogens, some serious pathogens that you would not want somebody to get hold of and be able to manipulate. How easy is it to get that coding? There was just a case recently, I think it's a Supreme Court case, or it's just an issue where the scientists who I think had discovered something in the genome of, whether it was influenza or another virus, who published but withheld information from a published paper that he felt should not be publicly available because of the dangerousness of it, the dangerousness of it. That he didn't want that being shared with, and there's real kind of turmoil in the science community that, on the one hand, that probably would be bad, but two, we need to know about that. Because what if somebody does actually get it and can do something bad with it? It would be good for the rest of the science community to understand it and be better prepared for dealing with it. So if anybody's going into law and policy, this might be a really interesting area to spend some time. I did mention it, but Ellen Jorgensen, the GenSpace woman, is part of something called the Personal Genome Project, which you saw Craig Venter on the What is Synthetic Biology piece, Craig, and then George Church at Harvard, and a number of others are real kind of leaders in kind of pushing the envelope of this whole area. Church and some others created the Personal Genome Project, which is 1,000 people who have agreed to have their DNA, their entire genome, sequenced and publicly published. And the motivation behind this project is to try to understand what all the implications of having human genomes available for whatever purposes, to try to stay ahead of all the implications and be better prepared. So, issues like if I had my genome done and my health insurance or life insurance company decided that they were, before they write a policy, want to see my genome. Ellen Jorgensen says the consent form that she signed to participate in this Personal Genome Project is really interesting reading. For example, one of the things, the number of things that you're warned could happen, one of which she said was, somebody could synthesize your DNA and plant it at a crime scene. And then on the other hand, you have this, the artemisinin and the jet fuel and all the biologics that are being produced that have the potential to do tremendous good. And like a lot of these technologies, they're neutral. It's this human use of them that really makes them forces for sustainability and good, or non-sustainability. Evil.
It's some of us were lucky to be at lunch with you earlier, and we were with the student. What was her first name? Allie. And she, for those of you who weren't there, she was part of the STEM project of last summer, which was an exercise like you've described. It really was a biohack. This was 8 students, was it 10? More than half a dozen mid-kids who came together and were said, figure out something to make that would be useful for the world. And so they ended up doing this. They ordered $800 worth of DNA, and then they've come close to a product that would, it's a luminescence type of product that would illustrate things that shouldn't be in water and so forth,
Yeah.
to keep it simple. So what I love about it, and I feel I really have learned in terms of our Center for Social Entrepreneurship, is this idea of hacking. You're not going to get groups and groups of mid-kids necessarily doing something like that, but the notion of pulling students together and saying, create something, is a great notion. And at one end, it is the high-powered biohacking. On the other end, it's just what do people need. In the middle is a kind of digital computer hacking, which is happening all around us. And it might be worth something for us to consider, you know, a kind of hackathon here, and see where it takes us. But that's the unifying thing I see. And then I think the great example of the airline company and so forth, in terms of, you know, what it means to students who are thinking about a life of social change and using this social entrepreneurship model.
Yeah. Yeah, I think it would be, you know, a field trip to one of these biolabs would be a pretty interesting experience.
Yeah.
There's some in Boston and Cambridge.
Right.
And, yeah, has anybody been to a maker's fair?
Yep.
These, they're doing it at Shelburne Farms every year, and it's, you know, the same ideas. It's people who are, you know, creating things, innovating, but actually building things.
Yeah.
I went to one of Shelburne Farms this past September, and it was fascinating. There was one table of about 15 people, all ages, you know, 7-year-olds to 60-year-olds, sitting there learning how to solder circuit boards. And you know, just guys in white lab coats, you know, and gals, just sort of showing them how to do this. There were people who were professional theater makeup artists who were, you know, turning kids into horrible looking zombies. Just and drones and, you know, all kinds of things. So it's the same spirit.
Yeah.
You know, let's try to, you know, hack a, you know, more sustainable product.
Right.
Or a more socially responsible innovation.
Okay.
All right. Well, thank you all.
That's it.
And then actually, while technically this is the end of the series, we are co-sponsoring on Thursday in the Wooden Colloquium up at Hillcrest a great talk from a really